Hello, my name is Cole Van Olen, and today I'm going to be going through a brief presentation on the parameters of soil microbial life. So this will look to discuss the factors that affect the range where life can be found, and then we'll go into looking at the high and low reaches where microbial life can be found in the soil profile. In order to get ourselves oriented, we're going to start off by looking at the global biodiversity as we know it today. So as it came out in 2016 in Nature Microbiology, they produced a new tree of life, which we can see here. And it might be a little hard to see, but it's not really the details that we want to get into. It's more of the overall structure and the different proportions that we see in this structure. So up top, the large portion of it, you see 92 bacterial phyla. In the bottom left, we have 26 archaeal phyla, and to the bottom right, you have 5 eukaryotic phyla. Now overall, with the information that was used to make this chart, we're seeing that over 2/3 of the biodiversity that's accounted for is found in microorganisms. Now, this is in part due to the new developments in high throughput molecular sequencing, and this has allowed us to gather a lot more organisms that we were previously able to culture on plate auger or other methods of that sort. So we have this vastly expanded portion of biodiversity that's found within microorganisms. And it's been said that the largest reservoir where they're finding these microorganisms is in soil profiles around the world. So we have this vastly expanding subset of biodiversity in this particular area that it's found in, the soil. So really, to get a better understanding of biodiversity as a whole, I think it's a good method to look into the soil and how it's creating this biodiversity. We can now look at the parameters that are at work in the soil profile. Now when we do this, we're looking at the range of these parameters. So it's similar to what you would see with Selford's law of tolerance, where we have a maximum and minimum values where organisms can exist, and then the middle range kind of that bell curve where it's optimal. So for today, the modulators that we're going to be looking at are temperature, pH, water, radiation, and particulate matter, which is a little bit harder to find, but one that we'll look at nonetheless. Now, when we look to understand these different parameters, it's important to note that when we're looking at them today, they define the limits of existence, but when you look at them over an expanded period of time, these same parameters are also the selective pressures that are driving evolution, speciation, and then henceforth that idea of biodiversity that we looked at previously. The first category that we'll look at for this is temperature. So if you were to look at a standard soil in the Midwest United States, the typical growing range would be from 18 to 24 degrees Celsius. Now, as we can see from this slide, looking at the far ends of that range, we find a -10 degrees all the way up to 75 degrees Celsius. So looking at the low side, the -10, this is from a permafrost that they think is between 20 and 40,000 years old in Kolomai, Siberia. So this Microbacter species 273-4 was found in a soil profile and was existing at a -10 degrees Celsius. On the other end of the spectrum, we see a high temperature of 75 degrees Celsius, and this was a Geobacillus thermovirens. And this was found in a geothermal volcanic soil environment off the Aeolian Islands of South Italy. and this is from a 2000 study by Romano. The next parameter that we'll look at is pH. So generally when you look at a bulk soil sample, you'll find a pH between 4 and 8.5 would be a typical range. Now looking at our extremes again, we see a pH of 0.7 all the way up to a pH of 10.5. So on the low end, we have a thermoacidophile archaea, which is the Picrophilus oshime, and this was isolated from a sulfuric soil in northern Japan, and it was near the area of Kawayu, and it was found near some sulfuric pools that are near the soil up there. And now jumping to our high sample, we see an alkalophilic psychrotolerant bacteria that was isolated from permafrost soil in the Tibetan Plateau near the region of Qinghai. And this sample was significant because the area that it's found in is a very high altitude but very relatively new alkaline soil. And again, this was around a pH of 10.5, and there's numerous micrococci families that were found in that soil. When looking at water content on the low side, we go to the Atacama Desert in Chile, and this is one of the driest and longest withstanding deserts in the world. Here they were able to find a hypolithic cyanobacteria that was existing as biofilms on pebbles in the driest regions of the desert. And this Crococci diopsis was able to exist there where regions see rainfalls, annual rainfalls of 2 millimeters per year or less. On the other end, there's less of a complete distinction as there's many soil profiles that can stay saturated for long periods of time. But this one example was found from Sopen Lake in Switzerland, and this is a methanogenic archaeon from an anoxic lake sediment. So this organism, this methanogen, was able to exist at a saturation, and it is Methanosacchina lacustris. The next modulator that we'll look at is radiation. Now we see some various distinctions when we look at the two ends of this. So on our low side, we find a nematode that was discovered in the Beatrix Gold Mine in South Africa. It was discovered deep within the mine, about 3.6 kilometers down into the mine. And so this nematode is Heliocephalobus mephisto, and again, found deep within the mine, existing on molecular processes there. On the high side, we find Deinococcus radiodurans, and this organism was isolated from the Sonoran Desert, and it can withstand radiation levels when isolated of 1,000 joules per meter squared, and this is very high, being that it's 4,000 times the dose that will kill a human. The final parameter that we'll look at today is the particulate matter that you can find within the soil. Now this can be very different for the different nutrients and toxins that you could find within a soil profile, but we'll focus in on just a couple today. So looking at the low side, we're looking at kind of a positive thing that you would want to see in the soil profile, and this is organic carbon. So going back to the Atacama Desert in Chile with the hypolytic cyanobacteria, coccidiopsis. There's an extremely low level of carbon there with 40 milligrams per kilogram of soil. Now, going to the high side with a toxin that would limit productivity in a soil, we go to the Dabashan mining area in China. And here they found and were able to isolate a proteobacteria that was resistant to heavy metals. So this mining area had extremely high levels of lead, zinc, copper, um, but this particular Burkholderia dabashanensis had a rough range of 278 milligrams per kilogram of lead that was existing in this soil. So this resistant proteobacteria was able to survive in that harsh environment. Now, in conclusion, in looking at these parameters, we see that soil microbiota can and do inhabit the far-reaching extremes of the globe. This plasticity has allowed them to fill vast niches, and in particular the soil. And at the end of the day, this gives us a better understanding of the drivers of biodiversity. And future research with these continuums that we're seeing in these different parameters will only take our understandings further. Thanks for watching, and I hope you enjoyed this presentation. This is a list of references from today's presentation, and if you found this presentation interesting and wanted to learn more, I would highly recommend Microbiology in Extreme Soils.